All right, Isabella, what we are working on, let me start with that. Um, they are working on, and Landon, you kind of disappeared on me too, so let me explain this to everybody. Actually, before I do that, let me get the GoGuardian going. Okay. All right, that's going. Okay. For those who were not here yesterday, on your to-do list, you should have a worksheet, and I'm missing the person I really need to see. Okay. But thank you all for being on time for class. I'm going to go ahead and get started with who we have. There they are. That's the one I need right there. Okay. All right. So again, if you weren't here yesterday, find the assignment, and um, there's a couple of things. Let's go back to looking at how I'm going to go back to the first assignment because I think that part needs to be, before we get to this part right here, let me go back to the previous assignment. So I'm going to go to home page and let's look at, oops, I think I clicked on the wrong thing. Nope, that's not what I wanted to do. I don't even know what that is. No, 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 no. Okay. I want to this. No, not letting me do it how I want to do it. But, okay, let me stop sharing and then I'll pull it up when I get the page pulled up that I want to look at. All right, so I'm going to start with the protein synthesis page because, like I said, we need to, some of you all need to go through this again. All right. And one of the questions asked specifically, what is the relationship between DNA, codon, and protein synthesis? So what that, and I'm telling you now, the answer to that question is they wanted you to say, and some of y'all are copying and pasting AI generated answers and it's not answering the question. What they wanted you to understand is that when you are translating DNA, the messenger RNA is going to copy the nitrogen bases and it's going to copy. And then when it goes back to the ribosome, the messenger RNA is read in sets of 3. So a codon is a set of 3 nitrogen bases. So that's what you needed to say. The DNA gives a message to the messenger RNA of the bases, and the bases are read 3 at a time. Okay, the bases are read 3 at a time, and we call that a codon. And each codon transcribes to one amino acid, and each amino acid that it transcribes for is then going to join to create a whole protein. So that's what this process is about. We are trying to create a whole protein. I did want to, so let me, now that I have it up, let me, all right, let me show this video instead for a second, because today's post. So if you understand, go ahead. If not, I want to go through this a little bit. And I started this video in the middle because I don't really need all the stuff at the beginning. But what we're looking at is the two processes of transcription and translation. So in the DNA, you have, and I'll let her speak.
In protein synthesis, we can look at two major steps. One is transcription, and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore, we're doing transcription.
Or actually, let me back that up just a little bit, because I like the way she said that. All right. So in that question on the worksheet for mutations, the relationship between DNA and RNA, this is the relationships. DNA, you can say, fetches some the message from the messenger, or sorry, the RNA fetches the message from the DNA, because the DNA is dictating a message. All right, so that's step one. All right, step two is it has to be transcribed. So it means it's not in a language that the RNA can tell the ribosome. So when it gets to the ribosome, it's got to translate it. Okay? And it's going to translate it. I'm trying not to show the whole video, but all right, there's a part that I really, all right, so we're getting there. So now we're at the ribosome. In the ribosome. Okay, I'm going to start from here.
For RNA, they carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important, because it's going to direct which tRNAs come in, and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't.
Okay, now notice that what she said, it reads them in threes. So what we're doing on the worksheet, by using our chart, we're looking at the three nitrogen bases that are in a set that is going to be transcribed by one transfer RNA. Okay, and there's an amino acid attached to that transfer RNA. It knows which one to bring because it's going to read the three bases in a row. Now, I'm going to take the caption off for now so that you can see. All right, now this is the part that's confusing some folks. You need to remember which bases are complementary to the other base. So in RNA, we don't have T. So every time, so now this thing that's inside the glob is our RNA, and the green thing at the top is the transfer RNA. There's an AUG is what the messenger RNA is saying. So the anticodon to that is going to be UAC, because A is going to bond with U, and U is going to bond with A, and C is going to bond or connect or is complementary to G. If this was DNA, excuse me, if this was DNA, all the U's would be T's, because T and A are complementary to each other. Okay, so this transfer RNA has a little amino acid on the end of it, and it's going to go look for the right amino acid to go with the code that is being transcribed for. Now, one of the things that you were supposed to look at in the first worksheet is that AUG is a start codon. It tells it this is where this, because you notice you have this long chain of messenger RNA. We're not reading all of it. We're reading a section of it. So it tells it where to start. Start at this section. So I have a triplet code that says start, and there's an amino acid that says start, and that's methionine. Okay, so it tells you start here, and then it's going to bring all the other amino acids together to complete the chain. All right. Okay. Looking at this screen here, you need to know what all of these are. So again, what is on the messenger RNA is called the codon. The part that's on the transfer RNA is called an anticodon, because it's kind of the opposite of what you first read. But the anticodon is needed to go get the amino acid. So the chart that you are looking at has the codon. It has whatever is on the messenger RNA. And so that's what we are reading when we do our chart. And then it brings the amino acid. So notice that methionine is up there. It connects the amino acid, and then the transfer RNA goes back to get another amino acid. So it's still coding for that particular one, but it's going to go back and get another one attached to it because it just dropped this one off. All right. And it's going to keep doing that. It's going to keep going down like a conveyor belt. All right. So when we're reading this chart, what we want to do, so it was, say, for example, it was our AUG. So we started with the A, and that's going to tell us that the answer that we're looking for on this box is in this third row. And then we want U. So we go up to, it tells you second base. You have a triplet codon, first, second, third. So the second base is U. So that means first column, second, third row. All right. And then we come to the third side. So we want to come down to that second row. I mean, that third row, because that's the row we need to operate in, but we need to come all the way back. So we want to find G, and we want to come all the way back over to the first box, because we need to be in the third row, second column, and we want the one that lines up with G from the third side, and that's methionine. Okay, that's methionine.
Corresponds with these codons.
Whoops.
I used to have a codon chart poster. Acid corresponds with these codons.
All right.
I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine.
Okay.
AUG is also considered a...
Whoops. All right, so there you go. So she just outlined how you're supposed to read that. And you read all of the codons this way. Okay? So every codon that was given to you on the first part of the worksheet, you had to read it on the chart and then come back and find the name of the amino acids that tRNA is going to bring back. So AUG is going to On the anticodon, it's going to have a reverse, but you're not worried about what the anticodon is. You're worried about the AUG because that's going to make this transfer RNA go get methionine. Okay, that's how that works. All right, let's see what else. They gave you a second example. Okay, right here. I'm going to stop right here. So this is a second example.
The complementary tRNA has the
Whoops. I wanted to stay here because it draws it out for you.
The complementary tRNA.
Okay, so the second one, we're trying to do CCA.
Has the anticodon
It keeps
G.
going. Stay there. Okay. All right, so again, triplets. So you want to say this, find the C in the first is the first letter, so that's the first base on the yellow side. The second C is going to be the second base is up here at the top. So automatically we know it's second row, second column, and then when we go to the third base, we know it's the third one, and in this case, all of them are the same. So there's 4 different ways to code for proline. That's what this box is telling me. I can have CCU, I can have CCC, I can have CCA, I can have CCG. All of those will code for proline. Okay? All right. Then we have this one over here that's going to tell us to stop. So again, we're not reading the whole chain. We are going to get to a segment that has a triplet code that when I go back to the chart, it's going to tell me to stop making this protein. So our little chain of amino acids that are connected at the top have been completed, and they will go off to form. Let me see if I can start there, I guess. All right, I'm going to let this finish out from here.
But remember, that mRNA was complementary to the DNA. So the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA.
And I'm gonna say that you could have done that as well. You could have said that for that question, what is the relationship between DNA codon and protein? You can say the DNA was the director, and it gives the triplet codon that is going to make the final protein. That's how you can answer that question. Okay. All right. Then another question asks you about
folding and
folding.
modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to.
So that was a quick review. All right. Okay. So again, that's how you read the box. That's how you would have answered this on this worksheet. And then using this circle, and again, I'm just reviewing for the sake of reviewing. The circle works the same way, except you start from the middle and you work your way out. So for example, with the C, I would have found the C at the bottom, this purple one in the middle, and the second letter is C. So I go to the second circle and I look at the circle of the letter C again, and I keep going in front of that for my third C. So in that beige circle, there's a C, and all of those again code for the same one. So it didn't matter which one I picked, and they're all going to code for proline. So proline is the answer I needed to write on the space. Okay. All right, now let's look at the let me do it from assignments because When I do it from the other page, it just goes to grades. All right, so the mutation exercise that you had to do worked the same way. All right, in the first box. So what they're showing you is now we are trying to make a protein molecule. This particular protein is hemoglobin. I'm sure you've heard of hemoglobin. If you've ever had your blood drawn, you know about hemoglobin. All right, so there's a special gene that makes sure that your hemoglobin is shaped the way it's supposed to be shaped. Okay? All right, so this first row is set up in the triplet codes for making. So this is the DNA, and the DNA is telling the messenger RNA what to do. So in that first row, we have C, A, C, and we want the complementary strand for the RNA for CAC. So again, we need, and if you need to put a little key on the side that sort of reminds you that every time you see a C, there's a G. Every time you see an A, when you are making RNA, every time you see an A, it's a U. Every time you see a C, again, that's a G. So the first thing in the first box, let me scoot this up just a little bit. So in the first box, the C, U, I'm sorry, the G, U, G is what you should have gotten for the first one. Okay, so then you come down to the chart. And that chart is going to, so if you start in the middle with the G in the yellow box, that's your first one. Then you go out to your second row, you want U, and so that's your orange one. And then the slice in front of that, you want G again, and again, all of those are going to code for valine. So that's going to be your first amino acid. It's going to be valine. Okay. All right, and this is for the benefit of those who are absent and those who are not doing the codons correctly. All right, our second one is GTG. Now, there are T's in DNA. There are no T's in RNA. So every time you see a T, it's still going to complement with A. It's just that there can't be any T's. Every time you have A, you have to have a U on RNA. So our RNA strand for GTG is going to be CAC. Okay, so we go back to our wheel or your box, whichever one of this particular worksheet has a wheel. So again, we're going to start with C, which is the blue one in the on the right side. Our second one is A. So when I go forward, I'm going to that green A. And see, this wheel is directing you which way to go. And then I'm going to go from that box. Now this one splits. Not all of them are going to be the same. So from A, I want C. So I want that purple box. And C tells me that this one, that CAC is going to code for histidine. Okay, so that's what I want to have for the amino acid on that box. All right, following. So you want to keep going all the way across on the first one. Now, I'm going to tell you that whatever you got for the first one, that's what the second one should look like, except when you get right over here to the end. Notice, I need to know you to notice that instead of CTT like we have on the first one, for our mutated one, we have CAT. So CAT is going to code for a different amino acid. So when this protein chain is made, it's not going to look like the first one. So therefore, it's going to tell this to, when the protein is made, it's going to create a different structure because it has different information to make something different. The first strand is going to, the question that I was referring to earlier, what is the relationship between DNA, codons, and protein? The DNA gives the instructions on what codon to make. The codons are the triplet code that is going to tell the transfer RNA what protein to make. And that's how you answer that question. And what protein it's going to make are these normal looking cells to the side here. So again, we're making hemoglobin, we're making red blood cells. That's what this particular chain of amino acids was instructed to do. Its job was to go make hemoglobin, which is a red blood cell. Okay. If it's normal, it's supposed to be nice and round, look like a donut. So that's what the first chain should have looked like. If it's our second chain is going to make it look like this. Now, folks, it's called sickle cell because we call this shape a sickle. Okay, so this is going to be bent. It's not straight. It's not round. It's curved. So we call it sickle shaped. Okay. All right, so it says, how does the shape of the protein relate to its function? So around, so then you need to go back and know what is the function of the red blood cell and how does that shape make it better to do its job than this shape, than the sickle shape. So that's what this question is asking you. First, you got to know what is the function. So you might have to look that up. Okay, because and I will just tell you the function of red blood cells is to carry oxygen. And we talked about that earlier, but for the sake of those who weren't here, the function of a red blood cell is to carry oxygen. So when we look at the normal shape, it carries more oxygen. The sickle shape cannot carry as much oxygen. All right. So, and it says refer to the details about this case. So that means we're not generating an AI answer to the question. We are looking at the pictures. We are looking at how this shape is related to the second shape, the sickle shape, and what does one shape have to do with the other as far as the function of that protein. And our protein is hemoglobin in this case. Okay, this is not a general case. Our protein this time is hemoglobin. Yes, I do, Isabella. Yes, I do. All right. Question 3. All right, question 3 is a fill in the blank. So you need to be able to tell a story in this paragraph. Again, the first part, the things in the brackets are not multiple choice. You are to give a name to what do we call ATGC? What is the name of ATGC? There are 4 of these things, and then finish the rest of that paragraph. Okay. That goes up until you get to when you say this shape is the molecule that determines its function. For these two down here, they go together also. So then it says changes in the DNA are called, and that was the whole point of this exercise. And in this case, which one are we looking at? And that's all you had to do with this one. Okay. All right, I talked a lot. There's only 10 minutes left to class. Hopefully that helped get this one worked out as well. If you have any other questions. So some of the ones that I graded, I made comments, and if you want to send it back to be regraded, please do so. All right, I need to unshare because I haven't done attendance yet. But if you have a question, please type it in the chat. I can check it right now. Okay. Other than when Ariana, when I responded to the last email that I got from you, did you send another one today? Okay. Yes. All right. So as soon as I do attendance, I'm going to go back and check the assignment. So if you um, submitted something or we needed to resubmit it, uh, I'll be checking as soon as I do attendance. Also, if you've already submitted yours and you didn't answer question number one the way I just explained it, you might want to pull that back and answer it again. Because now that I've explained it, and if you didn't answer it that way, I'm going to mark it wrong. All right, Malika, we good? I know I said I was going to do a pull-out session. Malik, you know how to correct your paper now? Okay, good. Okay, I will go check that right now. Okay. Um, oh, I'm so sorry. It should not be a 7. Where'd you get 7 from? Okay. What should that be? That should have been a 4. All right, that 7 should have been a 4. Not, I don't know where 7 came from. I guess 7 not even next to 4. I know I said 7. I'm looking at that, so that's my error. That should have been a 4. Okay, but I'm not. I'll deal with that. You fix it on your worksheet. It should be 4 because it should have been in order of 1 to 5, and your You still need to switch your 1 and 5. I did do that one correctly. Okay, so I will just do it like this. Okay, so it's been regraded and laid off of it. All right, folks, do keep in mind too, unless I take the late off of it, Canvas is going to start taking a quarter of a point off for every day that it's late. So out of the kindness of my heart, sometimes I take the late off so that you're not getting a 0. All right, so Ariana, sorry, not Ariana. Isabella, that one has been regraded. And again, if you would fix that on your paper, I know I said 7 on my corrected thing, but there was not really no 7. It should be 4. And then you want to fix your order so that it's 5-1, and now you have 1-5. That part I did give you right. I don't know where that 7 came from. Oh, okay. On the side number lock, the 4 and the 7 are next to each other. I do apologize for that. All right, that one is fixed. Okay, question 2. Question 2 basically is going backwards. So let me see if I can't pull that one back up. Question 2 is giving you the name of the amino acid, and you just have to search the chart to find that amino acids, and then you need to tell me what's in the box. All right, so for the first one, it said proline. So I'm going to look on the chart and I see proline is in the second row, second box. And all 4 of those triplet codes code for proline. So on that line, I want to write all 4 of those. All right, then we went to valine. So you want to find VAL. And here again, so valine, if I'm looking for VAL, that is the first column, bottom box. And VAL also has 4 different ways. So I want to write all 4 of those codes because there's 4 different ways I can get the amino acid for valine. All right, then histidines. I want to look up HIS, and I find that is in the second row, third column, and it is bracketed off. There's only 2 amino acids or 2 codons that will code for histidine. So I just want to write those 2. Okay. And then the same thing with GLY. Okay, good. I'm glad you got it. Okay. All right, any more questions? We got 1 minute left to class. Any more questions? I know reading these boxes are not the easiest, but that's how we figure out. You have to be able to read this chart in order to complete both worksheets. So now's the time to ask if you don't understand how to read the chart. Because these are all triplet codons on my worksheet. This ACT should be on the end. So hopefully on your worksheet, that'll be the end. Because ACT actually codes for, so if I come up here, I'm sorry, not ACT is the DNA. So sorry, you want ACT translates to UGA, not the college, but the amino acid. It translates to UGA. So I have U, G, and the A. UGA is a stop codon. So you want to just say stop on that first one. So ACT should code to UGA, and UGA is a stop codon, which means it's going to stop making that particular protein. All right, and the other one I think I would calling this GIN, it's actually GLN. GLN because it's glutamine. Okay. Any other questions? Questions. All right, time is up. And so we will see you all on Friday. We're going to finish up talking about mutations because this is the end of our unit. So when we look at this module, we're almost done with actually on Friday, we're going to look at technology and then review. So that's all we have to do with this unit. We're finished looking at technology. And again, this kind of goes back to the question that we did for our Halloween question. All right. Bye-bye. Have a good one.